Thank you. Good morning, everyone. It's always a pleasure to come here and speak. I think this is, although I always say that diagnostics is undervalued, I think we're at the beginning of a really exciting time for diagnostics, because with the announcement of all the funding by the Gates Foundation, the UK government, and World Bank and others, we do have the opportunity to really start something very sustainable and turning a completely new leaf in terms of diagnostics for NTDs. So I'm going to talk about the lack of progress, just giving an example, which I'm sure all of you know very well. And then I'm going to talk about the opportunities for improving the use of diagnostic tools for NTDs, mainly by leveraging advances in technology that have been driven by other diseases, big diseases like HIV, TB, where heavy investment into diagnostics have been made over the last 15 years or so. And then more recently, the threat of antimicrobial resistance, the call for a simple, rapid test to be able to direct physicians at the lowest levels of care to be able to prescribe antibiotics more appropriately. And what does that look like? And then, of course, all the global health emergencies that have driven the speed with which we need to develop diagnostics and validate them and make them work. And then I'm going to talk about some of the enabling structures that need to happen when we have the diagnostics that we needed for the different NTDs and the way forward. So leishmaniasis, this is a slide that I used when I was in WHO TDR in 2002 maybe. And so we haven't made much progress with that. We still heavily rely on the rapid tests to detect the antibodies to RK39, which is not ideal because antibody tests cannot tell you whether you have an acute infection or past treated infection. So it needs to be used in conjunction with a patient presenting with more than 2 weeks of fever and also an enlarged spleen. And so we really desperately need a very specific test. And if we only have microscopy, we need to use somehow think of not less invasive specimens than doing biopsies of the spleen or the lymph node. And we need a test of cure because the length of treatment is not been really well validated. When I was at WHO, I worked with the Chinese government on the use of diagnostics to think about how to use different types of diagnostics at different levels of prevalence for schisto. The Chinese government is very keen that since Chairman Mao eliminated schisto years ago by asking children as they go to school to pick up snails with their chopsticks and put it into a can, and when they reach school, hand it over to the teacher. Actually, that worked really well, and much of schisto was eliminated in China through that approach. But then with the Three Gorges Dam, the flooding, then everything re-emerged. And so the Chinese government wants to really look at how to eliminate schisto again. And so when you have high prevalence of schisto, you have high parasitic load, egg counts are high, and so you could use microscopy. But as you do rounds and rounds of mass treatment, microscopy is no longer sensitive enough. But you don't have a good antigen detection test because they're not sensitive enough. So the Chinese government started to develop antibody assays so that in communities they use antibody assays to followed by microscopy to know who to give treatment to once they get down to about 5%. And then the sentinel population is in school children using antibody assays. So And we said it would be nice if there's either a very cheap PCR or antigen detection assay before you start to use antibody. But again, that hasn't happened in spite of a lot of advocacy. Now, so what are the opportunities going forward? In the last 20 years, we advocated for assured diagnostics. So this is a term that I coined when I was at WHO to represent the kind of benchmark that we're looking for, something affordable, sensitive, and specific. And these are relative terms depending on the use of the test and what disease it is. And then user-friendly in that it could be done in a few steps. Rapid, giving the result within an hour or 30 minutes, robust so that it could be stored at room temperature and doesn't need equipment, or only you need minimal equipment that could be used with battery power or solar power, and then deliverable to those who need them. So essentially, when I talk to companies about having these a test with these characteristics, and all for 50 cents, they said, you gotta be kidding. You can't have all three. You cannot have things that are affordable, accurate, and then accessible. There has to be trade-offs. So what are some of the trade-offs in terms of accuracy? So I think that if you think about all the molecular tests, they are done in laboratories, and they are expensive and they're technically difficult, but they are highly accurate, right? And when you think about the other extreme with malaria rapid tests, HIV rapid tests, they're affordable, they're easy to do, but they're not as accurate as a lab-based test. So those are some of the trade-offs that you need to be thinking of as we select diagnostics or design diagnostics. So one of the drivers for technological innovation in diagnostics is HIV. And most recently, the UNAIDS and WHO came up with the 90-90-90 goals. And each one of those require a diagnostic test. So the idea is to have, by the year 2020, 90% of those who are infected with HIV should know their status. And then of those infected, 90% should be on treatment. And then of those on treatment, 90% should be virally suppressed. And so for the first test, for the first goal, you need HIV tests. And now there's a big movement towards self-testing that could be done in communities around the world by patients themselves rather than presenting to a clinic for care. And then once they're tested, then they could be linked to a care facility. The second one it used to be requiring CD4, but now we don't need it. So then the third 90 is for viral load. And so over the years, there's been a lot of funding that resulted in quantitative PCR assays being miniaturized and put into very, very small instruments that are sample in, answer out. So it's completely sealed, and the hands-on time is very short in terms of minutes, and nothing needs to be done. The result comes out on a computer or on a remote control for your phone. So these are the pipelines of machines that have been developed for HIV. And I've spoken to many of these companies, and they're willing to look at using the same technologies for NTDs, but there are certain guarantees that they need to make before they would invest millions of dollars into the targets. So one of the most successful, and is used for both HIV and TB, is the Gene Expert platform developed by Cepheid. And what it is each one of those ports is actually contains a mechanism to do an independent PCR. So it's completely random access. The company has developed a menu of many different targets that could be used. The first one they rolled out, of course, was detection of MTB with rifampicin resistance. And so you can imagine in a small clinic, if you have one of those smaller machines with 4 ports, in 1 minute you could put in a TB test, the next minute you could put in one for chlamydia, gonorrhea, the next minute you could put on one for HIV viral load or HCV, depending on the patients that come in. So that gives the real flexibility of being able to do many things with the same machine. And you can imagine that for NTDs that you could, depending on what NTDs are circulating in your area, you could have menus for that as well. And they're willing to speak to us about NTD, how to develop these for NTDs. And this year, they will launch the Omni, which is only 1 kilogram in weight, and it's about 9 inches tall. And contains everything that's in the big machine and could run everything, all the menu on the big machine. And it's about $3,000, just under $3,000 US. It's operated on a mobile device. It could use AC or battery. And so things are really moving forward in terms of being able to go to the patients rather than have the patients come to us. And all the quality control calibrations are done automatically through electronically. Now, with global health emergencies, I mean, we are experiencing epidemics of viral infections more frequently and more severe outbreaks on a sort of worldwide scale. And so I was in WHO during the outbreak of SARS, where we didn't even know what the disease is, let alone how to diagnose it. And there was a real scramble to not only identify the pathogen, but quickly make a diagnostic for it. And with viral infections, once they identify the cause of the infection, they could quickly sequence the genome, and then you could look at targets for molecular detection. And then it was a coronavirus, and then so that when we have MERS-CoV coming out of transmitted from camels into people in Saudi Arabia, we were very quickly able to identify the cause of the pathogen, the cause of the infection, and make a diagnostic test for it. But then, a year later, Ebola. We have diagnostics for Ebola, molecular tests for Ebola, but it takes about 2 hours long to do each one, and it has to be done in a safety isolation cabinet. And so you could imagine hundreds of patients waiting to be triaged to know whether they have EBV or not. And in fact, each day you could only do about 6 or 8, even if you work around the clock, you couldn't do very many diagnostic tests. So there's real need to speed up, to have a rapid detection assay. And then with Zika, we actually don't have really good serology that could be done on a population scale to know what is the risk of developing microcephaly amongst pregnant women, because we don't have a test that allows us to look at how many pregnant women have been exposed to Zika because of all the cross reactions with dengue infections in dengue endemic areas. It's much easier to tell in Europe, for example, when people haven't had a background of other flaviviruses. So with this WHO really wanted to look at how can we prepare for the inevitable, the next outbreak that's going to come. So WHO decided to have a call for open platform technologies to encourage private sector and public sector to work together and find these platforms that could be used at very short notice for new infections or new targets. And what can we do to get everything else ready so that when a new infection or new cause of outbreak is identified, we could quickly make a rapid diagnostic tests that are suitable for that outbreak without waiting for years for that to happen. And so I was asked to co-chair this open platform technologies progress, which is not only for diagnostics, but it's for vaccines, for therapeutics, and for reagents. And so this is still ongoing. WHO, in the first call, there were 35 applications, mostly from vaccine companies. There were 6 from diagnostic companies. In the end, 1 was selected to be presented to donors. And so we'll see whether these donors would be interested in looking at another round of these open platform technologies. Now, some of them, the technologies that are already available, I just want to give you an example of how simple and easy it is. So this is a layer I, which is a molecular test that actually can be, the whole assay could be done in about 20 minutes and 15 to 20 minutes, and requires only 2 minutes of hands-on time. Literally, you just put in the sample, cue in the information about that sample, and away you go. And there's cloud-based data storage. They're already approved for flu A and B, and as part of their influenza preparedness for Europe, they are doing clinical trials for chlamydia and gonorrhea and strep A, C. difficile, RSV, etc. And they will listen if we advocate for their platform to be used for NTDs. Roche acquired this technology from ICAM, and again, the results could be available in 30 minutes, and again, it was actually developed for flu A and B, and now they're applying it to HIV and HCV. The results are ready in 15, 20 minutes with this machine, which can be battery powered, and all the calibrations and everything is inside. Literally, you just have to put your sample into that sample holder, scan the barcode so that the machine knows which to run, and then put it into the machine. In response to the Ebola assay, bioMérieux actually have the BioFire film array available. And so now they've made it into a biothreat panel with Ebola and other hemorrhagic fevers. And all the reagents are dried in a pouch. And inside the machine, it does a 2-stage nested multiplex PCR. And again, it's very easy because it doesn't require any pipetting or all that. And hands-on time means only putting the result in, only putting the specimen into the machine. And also, again, they are responding to the call for more appropriate use of antibiotics by developing a respiratory panel that would allow physicians to know whether to use prescribe antibiotics for the patients or not. Now, I think I saw this displayed here at last year's conference, and it's recombinase polymerase amplification, which is developed by scientists at TwistDX in Cambridge, and has been used for different applications. And Amadou Sow, together with colleagues around the world, actually developed this suitcase, and so that it could be a diagnostic in a suitcase. And the left suitcase is for extraction, and the right suitcase is for detection. And it's pretty portable. And again, this is an open platform. You could design any pathogen on it. Now, with the threat of antimicrobial resistance, you could imagine that if a company claims that they have a test that can exclude all viral infections and include all bacterial infections so that you know whether to prescribe antibiotics or not, that would be a really difficult claim to think about how to include all the major causes of viral pneumonias and to be able to exclude all bacterial pneumonias. Years ago, we used to think that there must be some really good distinguishing features between bacterial and viral infections so that we could make this test. But 20 years later, nothing really has come out, except some host biomarkers. So now we do have incentives for companies and private sector developers to think about how to what kind of tests that could be used at the point of care at all different levels of the healthcare system to combat the threat of antimicrobial resistance. So the EU Horizon 2020 prize of 1 million euros has been awarded. I was on the prize jury last November, and there were some really good applications, but they were mostly host biomarkers. They were not pathogen markers. And so this is now a whole generation of new types of tests that could be used. But really, can you use one single host marker to distinguish between bacterial and viral infections? That remains to be seen. The test that won the EU Horizon 2020 prize, they said that will be in clinical trials next year. The UK Longitude Prize is still ongoing. We're trying to encourage not only UK scientists, but scientists around the world to enter into this prize. And there'll be funding for promising technological platforms, as well as some funding for discovery awards. That is coming up. So please go to the Longitude Prize website to look at that. And then the NIH has also got a prize for different types of point-of-care tests. So with that, we want to look at not only molecular platforms, but are there open serology platforms where you could detect whole signatures of either host proteins or antibodies. And there's the Luminix platform, which I think many of you may be familiar with, where the beads are actually color-coded with fluorescent dyes to produce a very specific spectral address on a microwell plate. And so with these beads and the tags that are attached to it, you could actually detect up to about 500 proteins or antibodies in a single well. And so you could imagine the power of that to complex all the different NTDs that are in your country, in your region, to be able to monitor your progress towards elimination. There are also a lot of nanotechnologies, nanowires, nanodots that do molecular barcoding as well that could be used. So with all these new technologies, what do we need? We really need each one of these technologies to have data transmission possibilities, because really our experience with HIV is trying to put in point-of-care technologies into HIV endemic countries in Africa, in spite of all the funding, because the health system is not able to, it's too fragile to have increased testing at all the different levels. The introduction of these new technologies actually puts a lot of stress into health systems, and in many cases they just simply break down, and the results are lost. And so we've learned over the years that we really need to strengthen health systems by having connectivity solutions. And these new machines all have data transmission possibilities, so that you can have real-time data on what's happening. And so you could use that to turn data into intelligence. But at the same time, it enables you to monitor the quality of the testing, the supply chain management, because each test result that comes in means one test has been used. And so, and then you could actually look at improving patient care. And we did such a pilot in Zimbabwe, where in one area, one region of Zimbabwe, we use a middleware solution to extract the same set of data from a GeneXpert machine, from the Alere HEMA for CD4, and from HemoQ for anemia, and have all the information be available to the Chief of Lab Services in Zimbabwe in real time, coupled with quality assurance data. So it can be done, but it needs to be done more generically, not for each company to have a specific types of connection. There are also serology tests that use the power of the phone to power the reaction. So this is developed by Sam Sear in Columbia University, where he has a microfluidic disk which could detect antibodies to HIV and syphilis. And the reaction is driven by the cell phone. And the cell phone can do 86 reactions before it needs to be charged. And the cell phone could read the results and transmit the data. So other than that, so with all these new technologies, it's now possible for communities to be more involved in testing rather than for everything to come top down. And so we looked at, the elimination of leishmaniasis in Nepal. And for Bangladesh and Nepal, they have done a tremendous job with elimination, and they're down to 300 cases from hundreds of thousands of cases. But for the Ministry of Health to be able to find those 300 cases is really challenging. So, so the idea is to borrow the example of leprosy, where women in the community are trained to look for skin lesions and refer them for testing for leprosy. So in the same way, the same approach, we're training women in the community to identify people with more than 2 weeks of fever that's unresolved to be referred to leishmaniasis testing, because the communities do have an interest in preventing the infections from coming back to the country. And schistocontrol testing for malaria in schools, those are some of these social innovations in health projects that we've identified through a call with WHO on innovation, looking at the power of communities to provide solutions for themselves. And in Malawi, they have transportation problems because Malawi is a very long country with only one main road. So many communities off the main road actually don't have access. So we've been working with UNICEF and one of the NGOs in Malawi to look at transporting blood HIV tests to those communities and bringing back blood spots for testing. And so these are many of the things that we couldn't imagine years ago, but it's now happening. So the way forward is that with all this announcement of new funding, hopefully we will get some of the new funding, is that we need to think about a plan for countries to integrate how best to use diagnostics to integrate as NTD control and monitoring, and also using the power of the new technologies and the data transmission possibilities to strengthen the healthcare system, to provide better services, better linkage to care, ensure the quality of testing, supply chain, and et cetera. And then involving communities in the use of diagnostics in case finding, and for monitoring progress towards elimination. These are really, really important, and we are, together with WHO, looking at new models of financing these technological developments and applying them to country programs. So thank you very much. And I have a lot of people to thank at the school and at WHO and for funding our work. Thank you.